Bismillahirrahmanirrahim. Today we are going to discuss the other types of the cells that are involved in immunity. Starting off with the B cells. As you all know, B cells, they migrate to the bone marrow during the adult life and matures there. They do not require the thymus for their maturation. If we talk about the lymph nodes, the B cells, they are seen in the germinal centers of the lymph node, while if we talk about the other important lymphoid organ, that is spleen, the white pulp of spleen that contains B cells. B cells, like T cells, like T lymphocyte, that is also present in the gut associated lymphoid tissue. If we talk about the major functions, they have two very important functions. Number one is they produce antibodies. Initially, the B cells, they differentiate into plasma cells, and the plasma cells, they produce the antibody. While the second important function is that they can present the antigens to the helper T cell. So this is the most important function, presentation of the antigen, and the second one is the production of the antibodies. B cells, they comprise of 30% pool of the circulating lymphocytes. The rest of the lymphocytes, they are the T cells. They are predominant in number. Maturation of B cells that occur in two phases. Initially, there is the antigen independent phase, and the second one is antigen dependent phase. The antigen independent phase, it comprises of the stem cells. From the stem cells, the pre-B cells are formed, and these pre-B cells, they give rise to B cells. So this was antigen independent phase. Now what happens in antigen dependent phase? The B cells, or the activated B cells, which are initially formed in the antigen independent phase, those activated B cells, they are converted into plasma cells, and it is from these plasma cells that ultimately antibodies will arise. So this is a brief overview about the introduction of or maturation of the B cells. One important point to be added for maturation is that when the pre-B cells, they are converted into B cells, an enzyme that is called as Bruton kinase or Bruton's tyrosine kinase, that is required. This Bruton kinase, it converts pre-B cells into B cells. So when there is deficiency of this Bruton kinase, it means that pre-B cells will not be converted into activated B cells, and ultimately no plasma cell that will be generated. So deficiency of this enzyme, it will result in a disease that is called as X-linked A gamma globulinemia. In this disease, the B cells, they are absent, and the person, he or she can suffer from severe bacterial infections like pyogenic infection. So this is important thing about the maturation of B cells, the deficiency of Bruton kinase enzyme or Bruton's tyrosine kinase. This slide, it shows how a B cell that is activated. If we consider that B cell is an antigen presenting cell, as we all know. So if B cell is an antigen presenting cells, numerous antigens, they bind to the surface IgM and causes the cross linking of adjacent immunoglobulin molecule. These immunoglobulin molecules, which have been cross linked, they aggregate and result in the formation of patches. These patches, then they move towards one pole of the cell, When they move towards one pole of the cell, it will result in the formation of a material that is called as capped material towards one pole. Because when when all the aggregates or all the patches they move towards one pole, the aggregates they will assemble towards one pole, they will, and the other pole there that will be vacant. So the pole that contain the patches that will mimic some appearance like a cap. So these patches move towards one pole of the cell and form a cap. When the capped material is formed, its endocytosis occurs. When the endocytosis occurs, the antigens they are processed and epitopes they appear on the surface, and these epitopes they are associated with class 2 major histocompatibility complex. This complex that is then recognized by a helper T cell, and helper T cell it will result in the secretion of interleukin 2, 4 and 5, and these interleukin they will stimulate the growth and differentiation of B cells. When we talk about the effector functions of B cells, the net result of B cell activation is the production of the plasma cells, and these plasma cells they will result in production of large number of immunoglobulin molecules. Plasma cells they secrete thousands of antibody molecules per second for a few days, and then they die. This is an important point to note. Thousands of antibodies they are secreted, and when they have secreted the antibodies, these plasma cells then they die. The activated B cells they also have the memory. These memory B cells they can remain quiescent for long periods, but they can become activated upon re-exposure to the same antigen or the antigen having more or less the same structures. So just like the T cells, the B cells they also have the memory. Next group of the cells that are present in immunity or that give protection and involved in immune responses, they are the antigen presenting cells. These antigen presenting cells they may include the natural killer cells, dendritic cells and the macrophages. If we talk about the macrophages, they have three very important functions: phagocytosis, antigen presentation and cytokine production. If we talk about phagocytosis, it is the ingestion of the microbes. When the microbes they are ingested, they are then killed by reactive oxygen species and reactive nitrogen species. So these two species, the reactive oxygen and reactive nitrogen species, they help in killing of these microbes when they are phagocytosed or when they are ingested by the macrophages. Similarly, lysosomal enzymes they are also helpful in killing of the microbes once they are phagocytosed. The second important function is the antigen presentation, and this antigen presentation it occurs in association with class 2 major histocompatibility complex, and this class 2 major histocompatibility complex it presents the antigen to CD4 positive helper T cells, and as you know this was the primary signal followed by a co-stimulatory signal involving B7 protein and B28 and CD28 protein. Ultimately it will result in the activation of T cells. Third important function is cytokine production. Numerous cytokines they are produced, numerous chemokines they are produced, the important one being the interleukin 1, tumor necrosis factor and interleukin 8. Next is the dendritic cell. If we talk about the dendritic cells, they are also called as professional antigen presenting cells or professional APCs. The dendritic cells, they are particularly important because they are the main inducers of primary antibody response. Why they are called as dendritic cells? Because they have long, narrow processes, and these processes, they resemble the dendrites that are present in the neuronal cells, and these processes, they make them efficient at making connection with the foreign antigens or the foreign bodies. Primarily the dendritic cells, they are located under the skin and mucosa primarily, and they at that place in underneath the skin, they are called as Langerhans cells. Dendritic cells, then they migrate from their peripheral location under the skin to the local lymph nodes, and when they migrate, they result in the presentation of antigens to the helper T cells. So dendritic cells, again, they are a type of antigen presenting cells. The third important type of antigen presenting cell is natural killer cells. When we talk about natural killer cells, they kill the virus infected cells. They have two very important functions. Number one is that they kill the virus infected cell, and number two is they produce interferon gamma. And this interferon gamma, it results in the activation of macrophages. How natural killer cells, they perform their function, you people have studied under that topic of CD8 positive cytotoxic T cells, that both of these types of the cells, the NK cells and CD8 T cells, they produce perforins and the granzymes. Perforin, they create pores or channels in the membrane, and the cell organelles, they are lost. Granzymes, they are caspases in nature, causes apoptosis. Similarly, there was a concept of antibody dependent cellular cytotoxicity that is also present here in the case of natural killer cells. Similarly, Fas ligand interaction, that is also helpful in killing of the microorganism, and this is also applicable for this natural killer cell. One very important point about NK cell is that they do not require the thymus for maturation and development, and like T cells and B lymphocytes, these natural killer cells, they do not have any memory. Neutrophils. Neutrophils, they are very important, and they usually come in our mind when we suffer from any kind of bacterial infection or any other infection. They represent the innate arm of the immunity or innate host defense. When we stain the cells, when we stain the neutrophils with hematoxin and eosin, the cytoplasm, it stains pink and it appears granular, while the nucleus, it becomes basophilic. And one important thing about nucleus of neutrophil, that its nucleus, it comprises of lobes. Usually 2 to 5 lobes are seen in the case of normal neutrophil. But if the number of the lobes of a nucleus, that is more than 5, that is called as multilobated neutrophil or hypersegmented neutrophil. And this hypersegmented or multilobated neutrophil, that is a classical sign of a very important type of the anemia, that is called as megaloblastic anemia. Neutrophils, they contain lysozymes. Lysozymes, they help in killing of the microbes. These neutrophils, they have receptors for immunoglobulin G molecules. They do not display any kind of MHC protein, but they have a very powerful microbicidal activity. And due to this powerful microbicidal activity, they act as a double-edged sword. ऐसी तलवार जिसकी दोनों धारियां तेज हों, double edged sword. On one side, due to its powerful microbicidal activity, it helps in killing of the microbes, it helps in killing of the bacteria. But on the other hand, it also results in tissue damage and tissue necrosis. So double-edged sword. Antibacterial, and on the second side, it also causes the tissue damage. So double-edged sword, that is very important for neutrophils. Eosinophils, they are also a type of white blood cells. Uh, these eosinophils, when we stain them with routine hematoxylin and eosin stain, or that is also called as right hematoxylin and eosin, they cytoplasm that attain a typical red color and granular cytoplasm. Why the cytoplasm of eosinophil that is red? It is because the eosinophil that is normally a negatively charged stain, while the cytoplasm of eosinophils, they contain a positively charged protein that is called as major basic protein. So this negatively charged eosin dye, it binds to the positively charged major basic protein, and this major basic protein that is present in the granules of this cytoplasm, it will result, or it will impart the red color to the cytoplasm. Eosinophils, they are particularly important in fighting against the parasitic infections, and their level that also increases in the type 1 hypersensitivity reaction. Another important point about eosinophils is that they display a weak phagocytic activity. Basophils, they are also a type of the white blood cells, and when we stain them with the routine hematoxylin eosin, or the right stain, that is also called as right stain, they appear to be blue. Why they appear blue? Because this hematoxylin eosin, it contains methylene blue, and this methylene blue that is positively charged, and this positively charged methylene blue, it binds to the negatively charged molecules which are present in the granules of basophils. So imparting blue color to the basophils. Two things are written, the third and fourth point. Basophils, they are present in the blood, while the tissue counterpart of the basophils, that is called as mast cells. Mast cells, they are seen in the respiratory mucosa and GIT mucosa, and also underneath the skin. And both the basophils and mast cells, they have the receptors for immunoglobulin E molecule, and that is the reason they are also involved in giving protection against the parasitic infestations and in the type 1 hypersensitivity reaction. So this was today's lecture, which was about the various types of the cells. T cells we studied in the previous lecture. Today we studied the B cells, the various antigen presenting cells, and various other types of white blood cells. In the next lecture, we will discuss the various cytokines and the important functions. Till then, Allah Hafiz.